Hello, my dear students. Welcome to my today's online class, that is continuation of recombinant DNA technology topics, paper 6. After having learnt the various aspects of recombinant DNA technology, now it is the time for us to learn the applications of RDT or applications of biotechnology as per your syllabus. One such application happens to be transgenesis. Now in this class we are going to study about transgenesis and its significance, knockout gene mouse production and knock-in mouse productions. Okay, how do you produce transgenic mice? So this is the quota for us. Let us see how much we are going to complete today. So this is first slide with my introduction. I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. You can see my cell phone number and email ID. You know why. After the talk, any queries, you are most welcome to contact me through any of the avenues. Okay. Now let us start the topic as such. What is transgenesis? Transgenesis, by definition, refers to the phenomenon of introduction of exogenous DNA into the genome to create and maintain a stable heritable character. So the definition says you are going to introduce the gene of your choice, that is exogenous DNA into the genome of the animal which you want to make it transgenic, so that once the experiment is over and you get a transgenic animal incorporating the gene of your choice, you are going to create and maintain a stable heritable character in that transgenic animal. That means once it starts reproducing, it will pass on the character of the DNA which you have inserted to the next coming generations. That's what the definition says. So I repeat, transgenesis refers to the phenomenon of introduction of exogenous DNA into the genome to create and maintain a stable heritable character. And you should know certain terms right now. The foreign DNA, what you are going to introduce, is called transgene. Okay, transgene. Then the animal whose genome is altered by adding one or more such transgenes is said to be transgenic. Say you have mouse with you. You are going to add the exogenous DNA through RDT experiments, and once the experimental animal gets this gene imbibed into its genome, then the transgenic mouse what you have produced is no more the wild type, but it has acquired one special character or two special characters depending upon how many transgenes you have introduced, and now the animal is said to be transgenic. We don't say wild mouse, we say transgenic mouse. Okay. Then the transgenes, once they are into the genome of the transgenic animal, they behave just like any other regular genes of that animal, and during reproduction they are passed on to the offsprings. That's what I meant by you are going to incorporate a stable heritable character in the transgenic animal. Okay. Then thus transgenic animals are genetically engineered or genetically modified organisms, GMOs. What is the speciality? They have a new heritable characters incorporated into them. How the new character came? By the gene what you have introduced. Okay. So this is what happens during the process of transgenesis. What is the significance? A favorite question of paper setters. Often they ask you to list the significance of transgenesis either for 3 marks or for 5 marks. It's a powerful tool for studying gene expression and developmental process in higher animals. It serves for the improvement of genetic characters of animals. It serve as good models for understanding the human diseases. Okay. Then several proteins of interest which have been produced by transgenic animals are of medical and pharmaceutical significance. So they have lot of medicinal importance, right? Then transgenic farm animals, not FARM, please note, PHARM animals. Transgenic farm animals today happen to be a part of lucrative worldwide biotechnology industry with great benefits to mankind. Okay. Then transgenic animals are important for improving the quality and quantity of milk, meat, eggs and wool production that normally the human beings use, which are of either food value or used for other purposes. It helps in creating drug resistant animals. So these are the significance of transgenic animals. After having understood the definition of transgenesis, after having learned few new terminologies, after having learned the significance of transgenesis, now we should learn transgenesis in mice. How are you going to produce transgenic mouse? Okay. Now, even today, mouse continues to be an animal of choice for most of the transgenic experiments. Why? Why means they are small, easy to handle and researcher friendly. Okay, it is easy to maintain the mice colony rather than an elephant in the laboratory, right? Therefore, they are researcher friendly. And they produces more ova. Okay, they produce more ova normally in one estrous cycle, that is the reproductive cycle, which the non-primate females practice rhythmically, that you will learn more about it in developmental biology class. So in one estrous cycle, a normal mouse produces 5 to 10 ova. That means potentially capable of giving birth to 5 to 10 pups, provided there is successful fertilization. Right. On the other hand, if you make them superovulate, okay, it is possible. I will tell you how they can be made to undergo superovulation. If you make them to superovulate, they can produce as many as 40 ova. Okay. This is the reason why again they happen to be the animal of choice. Thus, the methodology of transgenesis is well developed and standardized for laboratory mice and rodents. Okay. After knowing why the mice happen to be the animal of choice for our experiments, now you should know how are you going to produce transgenic mouse. Okay. You can produce by two methods, either by adding extra gene, then also it becomes transgenic, or by removing its regular gene from its genome, then also it becomes transgenic. If you introduce the exogenous DNA or foreign gene, then the technology is called knock-in technology. Okay, introduction of the genes is called knock-in technology. In contrary to the knockout technology, where you remove one or two particular genes to know their particular functions. Okay. First we shall study in this class how are you going to produce transgenic mice by knock-in technology. There are three methods for introducing a foreign gene into the mouse, and this is called knock-in technology. First method is retroviral method. Here you will make use of retroviral vectors. Retroviruses are a group of viruses which have been exploited to design vectors for RDT experiments. So these retroviral vectors can be made use of to produce transgenic mice by knock-in technology. So in this method, normally transfer of small pieces of DNA around 8 kb can be effectively carried out. But if your aim is to transfer gene of bigger size, this method is not useful. You have to go for other methods. I told you there are three methods. You have to go for other methods. This method is useless. So it is unsuitable for the transfer of larger genes. Okay. Up to 8 kb it's okay. Even for small genes there is a risk. There is a risk of losing some regulatory sequences which are very important for other processes, right? So there is a risk of losing some regulatory sequences. Then, apart from these two, there is risk of retroviral contamination, especially if you are aiming to produce food products which are often used for human consumption. Because of these limitations, this method of production of transgenic mice through retroviral vector method is not in regular use. Okay. Then the second method happens to be the micro-injection method. In my previous class, while discussing about physical methods of DNA transfer, I have taught you three methods: micro-injection, liposuction and electroporation. I hope students by now you are familiar with the micro-injection concept. Okay. Let's see what exactly is done in micro-injection method. The young virgin female mice. Young means virgin means the females which have not copulated with the males. They are called virgin females of 4 to 5 weeks age, okay, around 1 month, right? So the young virgin female mice are made to superovulate. What is this superovulation? Normally in one estrous cycle I told you they produce 4 to 5 ova, but you can always make them to superovulate in the sense you can give the hormones from outside source. This can be achieved by exogenous administration of hormones. You must be knowing a hormone called FSH, that is follicle stimulating hormone. You must be knowing another hormone called PMS, pregnant male serum hormone. Okay, mare is a female horse. Okay, pregnant male serum, right? So you can administer, you can give the injections of either FSH or PMS to the mice, and this has to be followed by exogenous administration of another hormone called HCG, human chorionic gonadotropin, after 2 days of administration of either FSH or PMS. So what you will do? You will take young virgin female mice. You will give the injections of either FSH or PMS. All dosages need to be fixed, maintained very thoroughly. Okay. And after 2 days, for these mice which have been already administered with FSH or PMS, you will give additional dose of HCG, that is human chorionic gonadotropin, after 2 days, so that the mice which have been treated with these hormones are ready to superovulate. That means instead of releasing 4 to 5 ova from their ovaries, they will release 40 ova. This is called superovulation. Okay. The superovulated mouse produces 30 to 35 eggs or even 40 eggs. Okay. So on one side you have the superovulated mouse. Okay. So which will release 30 to 35 eggs. Okay. Then, so figure 1 will depict the protocol of production of transgenic mice through DNA microinjection. What exactly is done? The superovulated mice are mated with males. Okay. The superovulated mice are mated with males, and next day, of course, in the estrus phase of the estrus cycle, otherwise it's a big waste. And then next day, these mice are sacrificed. That means killed. Sacrificed means killing. They are sacrificed under laboratory conditions, and after sacrificing, you can retrieve the fertilized eggs from their fallopian tubes, or what you say, oviducts. Okay. So first you will make the mice to superovulate, then you will make them mate with the males, and next day you will sacrifice them. Then you will cut open the fallopian tubes and you will retrieve the fertilized eggs. So you can see the fertilized egg here in the diagram 1 in the third step. Now for this fertilized egg, you will use microinjection technique, okay, where you will have a microinjection needle and a holding pipette. The DNA of your choice is introduced into the male pronucleus. Students, please note, in the fertilized egg you can see the 2 nuclei. The smaller one is the female pronucleus and the bigger one is the male pronucleus. You are going to inject the DNA of your choice into the bigger nucleus, that is the male pronucleus of the fertilized egg. But adequate care must be taken to ensure that when you are inserting the DNA of your choice, you have to puncture the elastic nuclear membrane, but at the same time you are not going to damage the nucleoli. Okay, you are not going to touch the nucleoli. The needle should not touch the nucleoli. For this purpose, if you want, a dissection microscope can be used for identifying the male pronucleus and for microinjection. Then, the eggs with transgenes are kept overnight in an incubator. You can't directly put it into the other mouse or the faster mice. The eggs with transgenes are kept overnight in the incubator, okay, so that they develop into a 2-celled stage by the process of cleavage. These eggs in 2-celled stage are implanted into a foster mother, ಬಾಡಿಗೆ ತಾಯಿ ಅಂತೀವಲ್ಲ ಅದು foster mother, that is pseudopregnant female mouse. How are you going to produce them? A pseudopregnant female mouse or a foster mouse is the one which has been mated the previous night with the vasectomized or infertile male. Okay? The foster mother can deliver pups after 3 weeks of implantation. You know that the gestation or pregnancy period in rats and mouse is 3 weeks. Okay? So please understand what you did. You will take the fertilized egg. You will identify male pronucleus and female pronucleus, and through microinjection technique, you will insert the DNA of your choice into the bigger nucleus, that is the male pronucleus. Then that you will incubate so that you will get 2-celled stage embryo. Now, on the other side, you should keep foster mothers ready. How are you going to do it? See, normally all rats, cannot implant the fertilized eggs artificially. They need to be kept ready. Okay, their uterus need to be primed up for the acceptance of the fertilized eggs. For that, what you have to do, they should get ready. That means they should be mated with the male ones, but the male ones should have been vasectomized. Vasectomy is surgical removal of vasa differentia, that is a sort of family planning operation, what you say in humans. So the males should be vasectomized, they must be infertile, they should not be able to fertilize the eggs though they copulate with the females. The moment the females are made to mate with the vasectomized or infertile male, their uterus get primed up. You know that in laboratory rats and mouse, you find bicornuate uterus to accommodate the pups, right? So, such females are called foster mothers or pseudo-pregnant females. They assume that they have undergone successful copulation and mating, therefore, they can carry the implanted embryos. So, it's a psychological status. So, such mouse are called foster mother or pseudo-pregnant female. And once you keep these pseudo-pregnant female mouse ready, you can always implant the 2-celled stage embryos into these foster mothers. So many you have to have. Then, one of these foster mothers, there are chances that they will carry the embryo which has taken the DNA of your choice. That means, after 3 weeks, you will get the pups out of these foster mothers. The presence of transgene in the pups. Right now, we will not be knowing in which of the pups you will have the gene of your choice. So, the presence of transgene in the pups can be identified by PCR technique, that is polymerase chain reaction technique you have in your syllabus. I am going to teach you little later. Or there is one more nucleic acid hybridization technique called southern blot hybridization, either through PCR or southern blot hybridization. You can know in which particular pups the transgenes have been incorporated. The transgenes are present. Okay. And then the mouse, so in the diagram you can see one pup with black color, just to show that is the transgenic founder. So the mouse carrying the foreign gene is the transgenic founder, this black one, okay, from which you can establish pure transgenic lines. Okay, this is how you are going to produce transgenic mouse by DNA microinjection method. Okay, another method I will teach you little later. Now, this method has its own limitations. So, here again I have described the procedure, that is superovulated males are mated with superovulated females, sorry for the spelling mistake, superovulated females are mated with males and sacrificed on the following day. The fertilized eggs are removed from the fallopian tube. Same thing I have taught you. Then my microinjection, you will incorporate the DNA of your choice. You will take enough care not to puncture the nucleoli, right? Then, so the eggs with transgenes are incubated. Then two-celled eggs are implanted into a foster mother, right? Then they will deliver the pups after 3 weeks of implantation. Then presence of transgene can be identified by PCR or southern blot hybridization. You can establish a transgenic founder from which you can establish transgenic lines. This is what I have explained right now. What are the limitations? First one, 3 to 5% of the inoculated eggs develop into transgenic animals. 3 to 5% of the inoculated eggs develop into transgenic animals, in the sense, if you have 100 animals in which you have incorporated the gene of your choice, you will get 3 or 5 transgenic mice. This is the rate. Okay. Then random integration of foreign gene into the host genome. That means the gene of your choice may not go and sit in the place where you want it to sit. Okay. So it may go anywhere and simply get incorporated into the genome. It has its own drawbacks. Then these transgenes, since they go and sit in odd positions, they may not be expressed, they may be underexpressed, or they may be overexpressed. Okay? Then the process is time consuming, costly, and labor intensive. Okay. So these are the drawbacks of this method of production of transgenic mice. But despite of all these limitations, this technique is routinely used for producing transgenic animals. So till now I have taught you the definition of transgenesis, few terminologies what you are supposed to know, and then the significance of transgenic animals, then transgenic mice, how to produce. Out of 3 methods, I have taught you 2 methods. Dear students, I am going to stop at this stage. In the continuation lecture, I am going to teach you the other method and knockout technology. Thank you for your patient hearing. Stay blessed. Thanks.